Welcome to the Learning and Research building where all the laboratory-based research happens on the second floor. Let's see what's cooking.
Hi, I'm Claire and I'm lead of the IGF's Metabolic Endocrinology group, and here we are on level 2 of the Learning and Research building. Our team is made up of postdocs, PhD students, master students and technicians, and our research focuses on how altered nutrition and metabolism impacts on chronic diseases such as cancer.
Most of our lab work begins here in a freezer set to minus 150 degrees Celsius.
Hi, my name is Karina and I'm a postdoc. In this minus 150 freezer, we've got hundreds of mobilized human cell lines stored and ready to be used in a whole range of research projects. These cells provide us with models for disorders such as cancers, and we can manipulate these models and look at the effects on the cells.
Those frozen vials of cells are taken to this lab, our cell culture lab. Here, they are defrosted and grown in an incubator set to 37 degrees to mimic body temperature, and with 5% CO2 levels to ensure that the normal physiological pH is maintained for optimum cell growth.
Hi, my name is Rachel and I'm a postdoc. Let me tell you a little bit more about our cells. We grow them in a single layer in these special flasks, and in here they have all the nutrients and conditions they need to survive and grow. Here is Anna, one of our PhD students, feeding her cells. In these cells we can observe the effects of genetic manipulations, such as knocking down or overexpressing a specific gene. We can also treat these cells with drugs, such as chemotherapeutic agents, and have a look at how the cells respond. For example, whether they become more or less mobile following these treatments, or if they continue to survive and grow, or if they die. Here is Susanna, one of our master's students, preparing drug doses to treat her cells with.
These effects can also be easily observed using a piece of equipment called HoloMonitor, as it records exactly how cells are behaving in real time. So HoloMonitor fits nicely inside of the incubator, and it captures the cells, shining a low power laser through them, giving you a holographic image, so there is no need for labels or stains. Here we have a video of our recent experiment where we can see how the cells are moving and dividing, and because HoloMonitor has a motorized stage, it allows us to simultaneously capture various treatments and even track each individual cell.
In addition to looking at how cells behave in response to altered exposures, we can also look at what happens to genetic materials and molecules within the cell.
Hi, I'm George. I'm one of the postdocs here at Southmead Hospital and belong to the IMEG group. In this lab, we extract DNA and RNA from cultured cells and tissues. DNA provides the genetic blueprint for all proteins required by the human body, whilst RNA plays a key role in turning this genetic information into protein. Join me now in the pre-PCR lab where we actually look at this information. Because the amounts are so small, we require a bigger quantity. This is conducted by a method called PCR, polymerase chain reaction. Once the samples are loaded into the plate, we take it next door into the post-PCR lab, where the plate is loaded onto the machine.
To look at changes in levels of proteins of interest, we can use a technique called Western blotting.
Hello, my name is Abdullah. I'm a PhD student. Here in Protein Lab, we are using the proteins that we cut them from our cells to separate them by a specific gel.
After separating proteins by size, now we will transfer them to a specific membrane,
which we can incubate with an antibody that will only bind to a specific protein that we are interested in. After the incubation process, we can visualize the protein on the membrane. Protein levels can also be measured by a method called enzyme-linked immunosorbent assay, or ELISA.
So for this method, we use these special plates which we can coat with an antibody that is specific for the protein that we want to measure in our cell samples. We then add our cell samples into the plate, and the protein of interest will stick to the antibody that's already on the plate. We then add a further antibody which is tagged with a colour label, and so the intensity of the colour that we see in our wells corresponds to the quantity of protein in our cell sample. One way of looking at proteins in tissue is by immunohistochemistry. To do this, we cut really thin sections of tissue and mount them on these microscope slides. These sections can then be labelled with antibodies specific to proteins of interest, which show up as a brown stain when viewed with a microscope. We can compare protein levels in the cancer tissue with the benign tissue from the same patient. These cohort studies are a great way of further investigating our findings from tissue culture.
In addition to our cell-based research, we are also participating in a number of clinical trials involving patients.
For these studies, we collect blood and tissue samples and can measure expression of gene and proteins in relation to a number of clinical and pathological features, such as if they relate to disease severity. We can also assess if they are changed by an intervention such as diet or exercise.
Hi, my name is Ella. I'm a PhD student in my last year. My project is about insulin-like growth factor binding protein 2 and how it is integrated with DNA repair mechanism in breast cancer cells.
Hi, I'm Abby, a PhD student in my second year, and I'm investigating novel biomarkers for the early detection of brain tumours.
Hi, I'm Zuzana. I'm a Master's Bioresearch student. My work is focused on breast cancer, specifically triple-negative breast cancer. I'm studying the role of estrogen receptor beta on breast cancer cell migration and invasion in the context of deregulated cholesterol metabolism.
We'd like to say a huge thank you to all Cancer Research UK supporters, volunteers and staff. Without your help and kind donations, none of this remarkable research could happen. Thank you.
We hope you have enjoyed seeing inside our labs and meeting our team, and hope that you have gained an insight in what we do. Thank you for watching.